Let me get a pointer here. All right, good afternoon. I hope you're all as tired as I am. So here's what I'm going to do. I'm going to try to you keep me awake right there. I keep looking at your bright face, and that wakes me up here. I'm going to try to blast through this one, and then if we can, I'll continue neonic, and I'll hope to finish with a neonic presentation now. So do you guys use KISS? Keep it simple, stupid? Okay, same type of thing. So as you probably imagine, I understand the genetics, I understand the PCR, I understand all that type of stuff. I'm not going to have any of that in this presentation at all. Okay, this is going to be a simple, a practical what you can do. And what I'm excited about right now is just stuff that I've learned in the last 2 weeks of this breakthrough, what I feel is a breakthrough on how quickly you can assess colonies for mite resistance, and that's the big thing. So traditionally, beekeepers have bred for bees forever. Back in Mesopotamia, 3,500, 4,000 years ago, beekeepers had already figured out how to select the best queens from their clay tube hives and propagate queens and do selective bee breeding. In Tel Rahaf, Israel, 3,500 years ago, they were bringing in queens from Turkey because they were a gentler bee rather than their native species of bee and breeding those. So bee breeding has been going on for a very, very long time. Beekeepers have known how to do this. The point of this presentation right here is if you look at all our different animal breeds and all of our different plant breeds, virtually all of them were bred without any knowledge of genetics before anybody heard of DNA. You don't have to know all this fancy whiz-bang scientific stuff to enter in a selective breeding program. So that's what this presentation is about, just to do a basic breeding program for bees. And forget all the big words. It's just a really simple process. I'm going to go through that. You have two choices. You got negative selection and you have positive selection. So negative selection is you don't breed from undesirables. So if you have a colony of bees that you'll take the lid off and it stings the hell out of you, you probably don't want to breed from that one. If the colonies die over the winter, you probably don't want to breed from the dead ones, okay? And you probably don't want to breed from the weak ones, okay? Then there's positive selection. If you have some colonies that make far more honey than the rest, then you may want to breed from those that make a lot of honey if that is your intent. So it depends, positive selection depends upon what your your income is, your income stream is. If your income stream is almond pollination, then you want great big bees early in the spring that build up. But if those bees then all starve or swarm before you make honey, then maybe you don't want a great big bee that builds up early. If you want to breed for bees that manage varroa on their own, maybe you don't want a great big colony that right after the winter solstice has a huge amount of brood. So think about your positive traits. These are all bred from the same mustard plant. Okay, all of our coals are all bred from the same mustard plant. So some of them you breed for white flowers, you breed for great big green flowers, you breed for little buds on the stems. They're all the same plant, all bred by people simply by positive selection, each generation breeding for ones that had certain desirable traits. Now, understand one thing though. Who was talking one earlier? Oh, I heard of some yesterday at the science thing. They kept saying breeding for a better bee, a better bee. The best bee is the wild type bee. That is the most fit bee in nature. So better is all relative. If you're breeding for more honey production at the sacrifice of something else, it may be better in your mind, but it doesn't necessarily mean it's a more fitter bee. Nature's already selected for the fittest bees for the trade-offs right there. And the reason is, in genetics, there's no such thing as a free lunch as there is not in anything else. I made this, I'll let you look at it just briefly. These are all the trade-offs that come up. For any trait you select for, there's a downside. So when you say a better bee, what do you mean by better? There's no free lunch. Every trait has a downside. And this is on my website. You can find it if you didn't finish reading. So let's look at the human have been breeding wolves for about 7,000 years, and we have reached the pinnacle of wolf breeding right here. Much better wolf, much better wolf. But would it survive out in the wild? Probably not. It has some pretty severe competition out there. So this is my point. When you say you're breeding a better bee, what is better? Okay? The other thing I keep hearing here, I heard it in Ireland, They won't want to breed any bee in England unless it's pitch black. If there's a spot of yellow, it's no good. And I keep hearing here is, oh man, we want the yellow bees. They breed for yellow. I don't give a shit in my operation what color my queens are. Yellow doesn't make me any money. Yellow does not make my beekeeping any easier anyway. Why would I waste good queens because they're quite some definition of pretty, okay? So you can do it if you want. I mean, if you're a queen producer and your market is yellow bees, well, go ahead, or purple bees or whatever you want. If you want bees with purple eyes, breed for that if it's your market. If you want bees that make you money otherwise or that survive otherwise, don't breed for pretty, breed for performance. So now what I want to focus on is varroa resistance. So now here's a bunch of traits you can breed for. You saw Eve speak this morning about some of the traits. So you could pull your hair out trying to figure out what varroa resistance trait you could breed for. The interesting thing is there's been several recent studies coming out where they let nature select by just simply doing this bond method, putting a bunch of bees on an island or at Avignon, France, or Norway, letting nature just select for the ones that survive after the rest die off, and they find out the traits that nature selects for are not the ones that the breeders have been selecting for, that the scientists have been selecting for. And the bees come up with things that we never, ever would have thought of. So the trait that I look at is, this was laid out way back in 1999 by John Harbaugh and Jeff Harris over at the USDA Baton Rouge Bee Lab. I put it there in yellow. Simply compare the growth of mite populations, okay? That's the easiest metric there is. So what I got here is the different, and they have a very nice paper with these R values, and typically our R values in most of our commercial stocks are around 0.020 right here. That's their intrinsic rate of reproduction, or increase of varroa per day while their bees are rearing brood. And there's some of them, you can get clear up to like a 0.030, and mites just eat those colonies right up. If you drop it down to these low levels, mites do not reproduce very rapidly in the colonies. So really, all I want is just to breed for those that keep mites down. The point is, consider your bees your employees. Define the job description and fire all those that don't meet it, and then promote those by doing it. And we do a very severe firing every year of those genes, because any queen who had headed a colony that didn't meet the job description, they don't make it to the next year. Only their daughters get promoted to the next year. Very simple. And to make it fair, I post this notice in every one of my bee yards so the bees understand what is expected of them. I don't want any complaints afterwards. So what you want to do is think about it. Think about what is really important to me. Do I want to waste my time throwing away half my genetics because they're not pretty and yellow? Or what is really the most important thing? What am I willing to deal with? We like gentle bees, make us money in almonds, make some honey, but mainly our main problem is varroa management. If we could just have bees that took care of varroa, that would make our life so much easier. Okay, here's a simple plan how to do it. First thing, start with a locally adapted stock. Okay, look at your climate and start with a bee that is adapted to your climate. If you're in a Mediterranean climate, an Italian bee would do well. If you're in a cooler climate, a carnelian type bee would do well. This is a map of North America. Each color is a different ecoregion. Ideally, and if we let nature run its course without us moving bees around, we would have a different race of bees living in each of those ecoregions. Nature would select for a race that was adapted for the specific flora that lived in that area, the specific pathogens that flourished in that area, for the duration of the winter, for the timing of the plant phenology. The bees would all be keyed in exactly to that flora and to the pathogens and to the temperature, humidity, the elevation, all those things. We keep moving bees around, but the recent research out of Europe, out of the Kolos group, shows the locally adapted bees do better than the bees brought in. For some reason, beekeepers always think the grass is greener on the other side of the fence. It's not. Bees that are adapted locally do best. The second thing is, I showed this slide the other day, is this difference of these feral bees. If you have a feral population, go remote from people where the bees are not just, the bees living in trees did not come from swarms, but actually have an established feral population. Those bees have already been selected by nature to be well adapted for that area. And if they're alive, that means they were handling Varroa on their own. So feral populations are very valuable. One of the points is it's unlikely, from what I'm hearing, for New Zealand to be importing any kind of semen of genetics. So even if in the United States we breed the most mite-resistant bee in the world, my guess is Australia will bring it right in, but maybe you guys won't bring it in. So that means you're going to be stuck with what you got here in this country already. So I would be really interested, if I were a New Zealander and being a breeder, seeing what's already out there in the feral population, the genetic reservoir that you have to try to breed from. Okay, so here's the whole concept of the entire thing. Really simple. You need to identify some metric to select the traits that you want, and then breed for those. The main trait I'm looking for is the colonies that have a slow mite buildup. So what you're going to do, you go out to your hives, or bring in whatever stock you're going to do, or capture those wild swarms, whatever it is. Run them in your hives. Pick out the colonies that you like the performance of those colonies in general. Okay, so you're going to negatively select for any that you don't like their performance, and then positively select for those that keep the mite levels lowest. And then from the best ones, and breed from enough that you maintain some genetic diversity, and raise a bunch of cells from those queen mothers. Then you're going to start a whole bunch of nuke or package colonies, all essentially even. All the same size, so that's an even start, because you're going to do this race, this competition between them. So start a whole bunch of them. Reduce the mite level down reasonably, so you need some mites to start with, but you want to essentially have them all start with essentially even mite levels. Then you're going to start them in a race for about 60 to 90 days and let those mites build up. You're going to put all those hives out in yards, let them mate out. You can't put, you're going to want to do this with a lot of hives. You're going to want to do this with 500 to 1,000 hives. So any small-scale beekeepers in here, you're not going to be able to do this, but any big people could do this. So I'll put them out. And the hardest things for you guys is in the Monica areas is to have an isolated mating yard. The reality is, if you don't have an isolated mating yard, you cannot do a breeding program. So you got to, at least for these, when you mate them, you got to find some place that's remote, that is that you don't have a bunch of other beekeepers sitting around. So it does not have to be a good honey production area. In California, we can just go to somewhere where nobody would, in their right mind, would put bees. And put your mating yard out there. They only have to be there for 2 weeks, okay? So you can feed them, keep them alive, let them mate, and then move them back to someplace where there's flowers or there's something desirable. The advantage that I have in my area is I'm the largest beekeeper in my county. And if you look at, if you Google map my locations, I control the drones in my mating yard. So that's a big advantage. And then you're going to start the race, the Varroa race. So everybody's on equal footing. Everybody's got a starting number of mites in the hive, and you're going to let those mites start building up, and the bees are going to try to keep their job description is to keep those mites from building up. So it's the bees against the mites now. And the mites are going to build up. And what I'm going to use is, instead of any kind of intensive opening the hives and dissecting combs or something else to figure out the mite level, simply look at the alcohol wash. And it's very predictable how fast that mite buildup will be at the alcohol wash. Then what I want to do is I want to do my first test during this buildup period, the 60, 90 days, something like that. My threshold for treatment for my hives is about 6 mites in an alcohol wash. If I get much above that, we start seeing virus issues. Typically, we start seeing virus issues when we get about over 15 mites in an alcohol wash in the middle of the season. So there's some latitude here. So what I want to do is I want to do my hold off my first treatment and my first assessment until the average mite level in these hives is running around 6 mites, and some of them will be up in that 6 and 20 range. But you want to build up enough that you can see a difference between the high and the low mite colonies. What we're going to do is we're going to look for the colonies that have 0s and 1s. That's the only ones we're interested in, the 6s and 15s. So we start monitoring with alcohol wash, and we sample every one of those hives all at just about the same time. The reality is what we're doing is, since we don't make all of our nukes the same day, we just keep track when we make our nukes, and then we're just following down. We're giving them all the same amount of time to build up, and then you do your alcohol wash. So if you're making 100 nukes a day, which is what we're doing pretty much, then we do 100 alcohol washes a day in the same sequence at the same period of time. So it's a doable thing. Now, those colonies that make the grade, that only have mites of counts of 0 or 1. Now, you may not have zeros or ones. If your mite counts are higher, anyway, you're going to go for the lowest mite counts. And you don't know what that is until you've already washed 20 or 30 colonies. And then you'll know what you're looking for. But in our operation, we're seeing, off of our better queens, we're seeing zeros and ones. Zeros, of course, is our favorite number right there. Okay, we're going to do the first selection right here. And then the ones that pass the first test, we're going to do a second selection before the mites get too high. And then if those that pass that test, we'll get to the third. Everybody else gets out of the program after the first test. Now here's the difference between this and the bond method, where the live and let die method. We don't want any colonies to die in this at all. There's no reason whatsoever to let any colonies die. If they don't make the grade, hit them with whatever miticide treatment you want. Just and put them into production. They're out of the trial. They're productive colonies. They can go on. They can make honey, do whatever you want. But you want those colonies to be alive because you want to have a bunch of healthy colonies the next season to make a whole bunch of nukes, but not with the same queens. You understand the concept? You don't need to punish the bees for not being mite resistant. You only need to punish the genetics, okay, and promote the genetics that are good. There's no reason to punish the bees and let them die. Treat them. There's no loss to you out of pocket for this. Selection is at the queen level, at the genetic level, okay? So this is what you're looking for. You're only going to select the winners are the genetics. Next spring, from those few now, out of that first 1,000, what we're looking at so far I've alcohol washed about 500, and I'll get another 5. The rest done the 5 days after I get back from this trip. I left the middle of our alcohol washing for this trip. So we'll do another 500 the next 5 days. Out of that 1,000, looks like right now we have about 100 to alcohol wash. So that means in another month and a half, I've only got to do 100 alcohol washes. And out of that, maybe I'll have 50, and I only have to do 50 alcohol washes before winter, and maybe I'll only take 25 colonies into the winter. So after your first alcohol wash, it's not a big deal. Your numbers are much smaller. Then the next spring, you rear a bunch of daughters from those few surviving ones, okay? And then you do something good. You go to all your numbers and you say, you want some free queen cells? And you just give all your neighbors free queen cells. What I went to my local beekeeping club, which includes most of all the other beekeepers in my county. And I said, how would you guys like it if I just gave you all free queen cells in the spring, and you guys put them in, and then your queens the next season will be producing all the drones, and I will control all the drones by giving away free queen cells. It cost me pennies to raise queen cells. I just give them away. Win-win situation. I said, how would you like to be in the first county in the United States to county-wide shift the genetics of our entire bee population to mite-resistant bees? They go, wow, that sounds pretty cool, huh? So my whole county, beekeepers have all bought into this. So I just give away the queen cells. Then you start the race again. Do the same exact thing the next year. That's it. How hard is that? Nothing hard, right? Now, there is one difficult thing. The cost. The cost of doing all those alcohol washes. And that's what I have choked on up to now. When people asked me last year, and the year before, they said, well, Randy, what proportion of your hives do you alcohol wash? Surely you can't alcohol wash them all. And I said, yeah, no way in the world I'm going to alcohol wash, 1,000 hives or 1,500 hives, okay? And now I'm saying, boy, was I wrong. Yes, we're alcohol washing. We're going to alcohol wash 1,000 to 1,500 hives this spring. I'm personally going to be doing it, okay? Because I figured out how to do it cost effectively. This is what you need. Or this is what I do. Here's my kit right here. I got a folding table. This was a recent, all this stuff, guys, has been in the last 2 weeks, okay? This is fresh, okay? It's been an incredible, I get goosebumps even thinking about it. I've learned so much these last 2 weeks. I'm excited to share it with you. And the folding table as we were going out, like on the third day of doing this, I said, oh, damn, I forgot. We needed a folding table. And my partner, or the employee I was driving with said, oh, we just drove by a yard sale. They had some folding tables out in the yard. Turn the truck around. Went back and bought one of these little folding tables. The tub, this is a Rubbermaid brand dishwashing tub. And Rubbermaid brand because that's the only brand of dishwashing tub I could find that the curve of the tub exactly matches the curve of my stainless steel half cup measuring cup. And so it saves me, it's all details. If you're going to do this 1,000 times, you want every detail to work. So get yourself a tub that has the curve that matches the curve of your measuring cup. Then I got my Might Wash plastic cups that I make. That's on my website. You make those things for a few pennies a piece. They're wonderful. First day we went out, we numbered them 1, 2, 3, 4. We found out, and it had matching blocks that were 1, 2, 3, and 4 to keep track of them, and didn't work. Too confusing because you keep trying to prioritize numbers. So next day we did an A, B, C, D. That wasn't any better. So I said, okay, what can we do? Pictures of fish and birds and reptiles or something like that. And I said, oh, how about color? So I just got some colored vinyl tape. Beautiful. The tape doesn't come off from the alcohol. You put it on little pieces of wood, and then we've got up to 5. We find 4 is all we need. 4 colored cups and 4 matching blocks is all we need. Then we go out, drive the pickup truck or the ute up to as close to the yard. Now, we've been doing it this last 2 weeks during a heat wave. The temperature's approaching 40 degrees. So, and my helpers, I don't want to kill my helpers. So I go out and do it out in the sun. I park the ute in the shade. So we've actually been walking the samples. But if you weren't in searing weather, you'd bring the ute right next to the hives. That would save you some time. And then I go out, pop the lid. We got all these colonies have built up to double deeps by this time. We don't have honey supers on these, but if you had honey supers on, you just go down to near the brood nest, and you pull out a frame essentially the same. So I just look down for where the cluster is, and I just want to be in the cluster kind of towards the edge of the brood nest. You want to sample essentially similarly. Any frame that's got any bee bread on it next to brood will do fine. There's plenty of nurses, and you've already seen how to do this. Now, pick it out. And usually I have my table just a few steps from the hive. And as I walk there, and I can do this, I can walk and look for the queen at the same time. Now, I tried it also. I cannot walk, chew gum, and look for the queen at the same time. So I cut out the gum chewing, just walking and looking for the queen. So you walk towards the, first, there's hardly ever a queen on that because you didn't pull the frame from the brood nest, okay? You walk over to the tub. One other look. Now, all you need is one experienced person who's able to spot queens. In this case, it's me. The other person doesn't even need to be a beekeeper. Just some technician who can actually do something accurately. So one person, we typically do a 2-man crew now. We've tried it up to 4 people, and 4 people we're down to a minute a wash. With 2 people, it's 2 minutes a wash, okay? But 2 people is plenty. One experienced, one experience. Look for the queen. And at this point, you also do your negative and positive selection for other traits. You can look at the brood pattern and see if they're how they're doing. See if they're stinging you. Find a nice brood pattern. Yeah, that looks pretty good. I like that one. Okay. See if they're whitening wax when the other ones aren't. See if that's an indication of their ability competing in the same yard, whether they're going to be a good honey producer. Check carefully. If you see the queen right there, pick her off, put her back in the hive. Okay, now you can shake those bees off. You don't have to worry about finding the queen at all. Then one little quick hard snap shake. No more bees than that. Any more bees than that, I just shake them down, take the cup, scoops them out, and put them back in the hive. You only want enough bees in there just that you can get a half cup measuring cup. If you have any more bees than that, it's hard to look for the queen in there. Now we do the usual incantation, ask for volunteers, the old bees fly off, that kind of stuff, okay? There they go. I just told them. Old bees fly off. They all come out of the tub. The remaining bees spread out into a thin layer, and it's just really easy to look in there. If the queen's right there, she just stands out like a sore thumb. Same thing. Put her aside then. Then I shake all the bees down to one side and just kind of tumble them into the cup. I don't do so much scooping. I just tumble them into the cup. And as I'm coming out, I just go with my finger like this, right across the top. And I've done this thousands of times. I've never been stung a single time. You just scoop them off the top of the cup because you want to get in the same even amount. Dump them into the alcohol. Snap the lid on. And then notice I have the blue marker next to the blue cup. You snap the lid on, and then your hand goes right to the marker. And when you go tap the rest of the bees into the hive with the tub, put the lid on, you put the marker on that hive. We also found I can also just do with an inkjet printer that the ink does not dissolve in alcohol. So like for the controlled trial we're doing now for EPA for the oxalic acid, I have inkjet printer labels. So we actually have we're using the color plus an extra validation of a hive number. So if your hive numbers are numbered, you can use that. The other thing is, when I saw the presentation this morning about the lady who was keeping track of all your inspections for your AFB, I said, you already do this? You already open every hive? This would add 30 seconds. 30 seconds per hive. It would be nothing for you to add to do this. Okay, so if the ute's right next to us, we just hand the cup over. If the youth's parked in the shade like it is here because it's so hot, a helper comes out and ferries the cups back and forth, 2 or 3 at a time. Oh, I didn't show you this. In my table, I drilled a couple of holes so the cups just fit in there. So when I move the table, the cups don't move over and they don't spill out. I need to modify this. I want to have all 4 holes. Right now I got only 2 holes, but 4 holes. A 3-inch hole saw just makes the perfect size, and the cups just drop in there. Saved me a lot of trouble. Okay, so this day I had 2 helpers, Tara and Rachel. Tara's worked with me for a long time. She knows just exactly what to do. And it's pretty warm, so they dress pretty lightly on those days. There's Tara with the whole setup right there. So there's the mite washer right here. She has a white plastic tray right here, so you have a white background. So in case you spill a cup, you don't lose all the mites or the sample. You can just wash it right back into there, and you have a white background, so you can look down and see the mites very easily. And that keeps everything in a nice, clean, tidy area, and you keep it wiped down with a towel. And plenty of alcohol reserved there. And what we do is, you can see right here, we put everything in these half-gallon canning jars. These are the same jars we use for our syrup feeders. And they fit, the mouth of those canning jars fit those cups very nicely. They just drop in halfway. So it's a real simple system with these the jars. Okay, so we bring them over. We have the mite washer. It's vibrating like this. We never turn it off. When you get to the yard, you turn it on, and you just pick the cups in and out. And you rotate them in a pattern as you put them in. So if you're rotating a few, they wash for 60 seconds very, very easily. That gets 100% recovery of mites. And I've tested this a couple of times where I've had them dump all the bees into a bucket, and we get two gallons of dead bees. So that's 8 liters of dead bees. And when I've re-washed them, one time I recovered 0 mites, and one time I recovered 1 mite. Okay, so we're getting 100% of the mites on these washes. Okay, so the sample comes in, sets it down. She takes the lid off. She takes the bees out slowly, and she sets that cup with the bees into this cup, and the alcohol drips off. We don't need to see those bees again, okay? They're going to wind up in the compost pile or tossed on the ground. We want this cup right here. And then the mites have all just sunk like rocks to the bottom of the cup, so you just look at the cup and you just pour it off, and you pour the alcohol back into the alcohol jar for reuse. We just keep reusing that same alcohol all the time. You don't have to strain it or filter it at all, because the mites are all at the bottom. But we do. We just run it through a little tea strainer just to make sure we don't spill anything. But no mites get spilled. And then she looks down through the cup, counts the mites, writes it down on a card, and then when she's done, she swirls the cup, pours the alcohol and the mites out, strains the mites out, and the alcohol goes back in the jar, and that alcohol just keeps getting reused over and over again. Then we let those, at the end of the day, we let the jars of alcohol sit overnight. Any particles in there just settle right to the ground, and we just decant the clean alcohol the next morning, and we're off again using it. We just keep topping off the alcohol with fresh alcohol. So you don't even go through that much alcohol. Very easy. Don't try running the alcohol through any kind of filter. Just a strainer and let it settle. Filters just clog up. It's really hard to do. And then here's Rachel carrying a couple of samples back, and in her hand, she has the 3x5 cards to mark the cards, has the hive notes. And she goes back with the color-coded cup and the card. She looks for the hive that has that color code. She picks up the lid and puts the 3x5 card in there. Now we have the information we need on there. And if there's any notes, like I want to say, oh, this colony is queenless, this colony has this problem, I say, okay, mark the blue this, mark the green this, and that note is on there for the next person to come through. And there's one with a mite count of 11. There's one with a mite count of 60. We're counting zeros. We're getting wide spreads. When you get that kind of spread where you're selecting zeros against 60s, you've got some selective pressure going there. We're weeding those big ones out of there. And there's Rachel happy with a 0. So any one that's a 0, we write breeder and 0 on it so that colony does not get treated. This is our typical for 2 people. 30 hives in 40 minutes, complete. It is fast. I've done this for enough yards now. It's amazing. Oh, why is that not showing? Okay, it works out to cost us, when I figure my time at a high price and the helper's time at a low price, it's about $1.50 a yard, $1.50 a hive. If we were doing typical labor price for 2 low-paid people, it would be $1 a hive. Okay, that's US dollars. It is cheap to do this. It's cheaper than most of our mite treatments. And what we're finding is I can skip a treatment in an entire yard. I can save myself a whole lot of money for just a few dollars for labor. And we know everything we need to know about every hive in that yard. And now these are histograms from just several yards. So what this says is, in this yard, we had 3 colonies that had zeros. We had 5 colonies that had a mite count of 2, 3 colonies with a mite count of 3, 1 with a mite count of 44, and a couple of 28s. So out of that yard, well, like this one up here, now we know, here's our breeders, there's our mite bombs. The mite bombs, when the crew comes through later, they're going to pick up all those mite bombs, and they're going to bring them back home, or to a sick yard, split them in half, blast them with formic acid, kill all those queens, start those fresh right there. Now, the bees are still fine, you just put a fresh queen in there, and the breeders are all going to be moved to a breeder yard, and everybody else is going to be in good shape. So I'm offering this out to the bee industry. This is a very simple, inexpensive way to get into a serious selective breeding program. If we all were to do this every season, varroa would be a non-issue. Any questions? Okay, I'll tell you what, I'm going to finish with the neonic. You want to hear the rest of the neonic one? Yeah, go ahead.
What's the percentage of the alcohol you're using for the alcohol wash, and how long can you reuse it before it dissolves too much to be used further on?
We start with 70. Well, it depends. Actually, we were using 70, but at the dollar store, we can get 50% really cheap. And we just keep reusing it. What we just keep doing is, in every wash, in those bees you take out, they take some alcohol with them. So all day long, you're adding a little bit of fresh alcohol in there. So we never ever toss any alcohol. The alcohol is just used indefinitely. Yeah.
Yeah, Randy. What's your long-term trend looking like? How many years have you been doing this? What have you?
This is our second year of doing it. So you asked me that in about 3 months. Okay? So I'm knocking on wood that it's looking good. But what we found last year, it looked pretty good. The ones that we picked out early that were zeros, again, we lost about half of those at each subsequent testing that went up. But at the end of the season, we still had some zeros. Okay, so I don't know if we'll be able to pull it off again, but they were sure hoping for it.
So, in your opinion, then, it'd be a good investment in time and money to go through a commercial operation of 1,000 or 2,000 hives, might wash everything at the same time of year to find the low mite levels. If
you're interested in breeding, and actually what I'm finding out, if your treatments cost you very much, it could save you a ton of money in treatments too. So like I said, if you would have asked me this 6 months ago, I would have said it's impossible. But like I said the other day, impossible is only as good, only lasts until everybody's doing something. Then it's just normal. So impossible shifts to normal really, really fast, okay? Good, I got done early here. Okay, no, that's snipping tool. Come on, where's my pointer? There we go. Come on. Okay, it's not cooperating. It's going to be over. Oh, I know why. Because I wasn't running it off of this computer. I was running it off of, I need to reopen the file. Recent, New Zealand, Neonics, this one. Okay, and we had gotten down to, yeah.
If you've got time, would you be able to just verbally just run through that system in a sort of a sentence each step so we can pick up on it again? Because, flashing it through there, it's pretty hard to keep up with what you're proposing there, or have you got it on your website?
Are you saying I talk fast?
No, nothing to do with that, no. I wouldn't possibly suggest that.
That's okay. It wouldn't be the first time I've been told that.
Yeah, but is it on your website, or is it so new that it isn't on your website yet?
No, this is fresh as of this morning. No, this is...
That's what I thought.
Yeah, this is a very up-to-date... I will be publishing this soon. Which you wanted to see the first part of this, or where are you? There you are. The first part or the last part?
No, just that process that you went through.
The process, okay.
Just in summary, if possible.
Oh, don't you want to see the picture of the queen bee again? I like that one. We had a, whoops, go back here. What's interesting, we had a queen producer in California, a well-known queen producer, and got a ride to Italy because they produce Italian queens to pick some of the genetic stock from Italy. And when they got there, the Italians said, "Oh God, this is the best Italian queens, this producer, famous." So they went over there to get that, and he goes, "Oh yeah," and she showed this slideshow, and I watched her show it to 2 different groups, and he pulls out this frame, here's this queen, she goes, and this the most famous queens in Italy. She goes, "But there was a speck of black on her abdomen, so we couldn't take her." Went all the way to Italy to get the best Italian stock, but there was a speck of black on there. So out of the program, okay? Okay, oops, let's see. Okay, bunch of queen cells. Hang on a second, why is this doing this? Oh gosh. Come on, Microsoft. No, that's the wrong one. I just learned how to do this, hang on. I right click here, and I go to settings, and I say automatically hide taskbar on desktop mode. And that should disappear, that taskbar. Damn. Oh well, we still got the taskbar on the bottom. Okay, so make a bunch of nukes. Or make a bunch of queen cells. Make a bunch of nukes or packages. Even better is to do it with, if you really want to be scientific, you shake all your bees into a great big box and then let them homogenize and then take them out and start with packaged bees or shook bees. But we just do it with nukes. Equalize the mite levels. Put them out into mating yards, and then if you need to, bring them back to a yard to build up, but we're able to mate them and let them build up in the same yards. We got about 50 yards of them. You start the race. After about 60, 90 days, when the mite levels get to the point where you would normally be treating, you may even want to push a little bit on that. You check them for mite count, and then you're going to repeat that test probably 2 more times during the season. Don't need to let any colonies die. Go ahead and you treat all the rest. Okay, that next season, you rear daughters from the best. Oops. Okay, so here we go. So mite wash, colored cups, colored blocks, The little table is a big one, and the holes in it are also big ones because nothing spills then. Pull out the frame. Make sure it's near the brood nest. Check for the queen. Check your brood, your honey, your gentleness, anything else, other traits that you want, any kind of disease. Anything with a disease is obviously not going to be in the program. Look for how well they're comparing to the other hives for honey production. So we put a star and say honey on the cards of the ones that are good honey producers, because if we have our choice of breeders, we'd rather have the breeders that were good for honey production. Look for the queens. If you see the queen, just put her back in. Shake them in. Let the bees, only about that many bees, let the rest of them fly off. Look for the queen again. If you see the queen, stands out very easily, even if they're not yellow. Scoop up the bees, level them off, into the cup, snap on the lid, pick up the block, put the block on top of the hive, mark them with number if you wish, send the cups back to the truck. So there's 2 or 3 trucks going on, or cups on the truck. Normally the ute is much closer to there, but the girls were just dying from the heat, so I didn't want them to take any more clothes off, so I moved the truck off into the into the shade. Okay, and then here's the wash station, and like I said, normally it's just Tara and me, just a few feet apart, so it's just really, really fast. Whoever is ahead is the one that's moving the cups. There's the wash station. There's the spare alcohol for topping off. We start off with each of those jars is 2 liters, so we start off with about 4 liters to start with and have backup alcohol, and that's plenty for us to work with. And everything fits. Every jar fits. Every hole fits. It's all in deep. If you want to do something efficiently, you eliminate any chance of screwing up. Make sure that everything fits together. And it just so happens that these wide-mouth pint mason jars have the same lid size as the big jars, and that size, I drill that same hole size in the table, and everything just works beautifully. Okay, when it comes back, she washes them, lets them go in the washer right there, pulls out the bees, let them drip off, let the alcohol drain off here so we don't lose so much alcohol. And the amount when we toss these bees on the ground, that's your alcohol overturns. Enough removed each time that your alcohol doesn't get diluted too much by the fluids from the bees. Oh, I didn't put in? You don't want to do this during a honey flow. And if you do during honey flow, because the bees get sticky, you got to have your water bottle. I got to add that slide in there. And you need to wash out this tub about every 5 or 10 colonies, because if it starts to get sticky, it's a problem. But if they're not shaking nectar, it's much easier. So you pour off the excess alcohol, looks for the mites in there, then swirls it around, pours off the mites, the alcohol. Look in the cup against the white background, make sure there's no residual mites in there. But once you get the technique down of the swirl and pour, there's very rarely a mite left in the cup, unless you get like those 60s and 48s. Then usually those have to be rinsed a second time. Alcohol settles down overnight, and before you take off in the morning, before you jiggle it on the truck, you pour it off clean. So you can see this alcohol has been used a fair amount. This one's a little bit fresher right there. Carrying the cups back with the note, color-coded there. But if we were really good, we'd probably have color-coded 3x5 cards. That's a good idea. I'm improving this method as I'm talking, okay? So you go back to the right hive, and then you pick up the block when you put the note down, and that goes back with the empty refilled cup back onto the table. So there's no, we actually, this works really good. There's like close to 0 screw-ups on this. Once you get it down, and you smile when you get the zeros, the ones that have mite counts of 0. Very quick. Takes us on the average, the 2 of us, it's taking us 2 minutes per hive in a yard, and just a couple minutes for setup and breakdown. Then we're off to the next yard. How's that? That good? Okay. Now, back to neonics. Where were we up to? We were up to spurious correlations, I think, from current slide. So you all got the warning about cheese consumption and being strangled in your bed sheets, okay? I probably saved lives right here. Yeah, this was a real interesting one. How much time do I have left? Anybody know? Oh, God, I can slow down. Okay. What I did is they said there was these correlations between butterfly decline and neonicotinoid use in Sacramento. Well, Sacramento was just the next county to me. So I just went on the Sacramento Ag Commissioner's website, and I downloaded how many acres were planted to anything that would have had a neonicotinoid on it. It worked out to be about 4% of the acreage of the entire county. And then I went to the California State Department of Pesticide Regulation, and you could download pesticide use reports, so I could see every application that was made of any neonic in that county during that year. And I found out there was virtually no neonic use in that county, just on a few percent of the acres, hardly enough to cause the decline of any species. And then when after I corresponded with the authors challenging them, the lead author referred me to the senior author, and he was the one who said, well, I really didn't want this paper to get published anyway, but we did it, and I see this a lot. Here's the deal. When a doctoral student is working on their dissertation and they come up with a research project, they often don't know anything much about what they're going to be working on, okay? They're on a learning curve. And so they do an experiment design, they start this big experiment, and a year into it they realize, oh God, did I screw up? The protocol was terrible. This is, a waste of this, but they got so much work in it, they say, well, we'll go ahead and we'll take it to fruition and we'll publish it nonetheless. I see this all the time, okay? So be really careful if it's a new researcher who has published something. So it was published, but again, this guy said these butterflies were declining for 10 years before the neonics, and it only showed any kind of correlation in one county or two counties out of three. Now, the other thing is, yes, there are problems with the neonics. If you treat a one-frame nucleus colony with neonics, it will crash. There's big effects on the queen. But there's a buffering effect of the bees. The larger the colony in the honeybee, the less of the effect. You start getting up to a 5-frame nuke, there's very little effect, and by the time you get to a 10-frame colony, you don't see an effect. So there's clearly an effect, but it only seems to be in very small colonies. That's maybe have been what happened here with Jeff Williams' study. Jeff's a friend of mine, so I challenged him and I said, hey, I'd be happy to replicate this in California because I have a hard time believing this one, that there was that strong an effect on the queens. What he did is he fed colonies continuous pollen patties spiked with neonics and then used those bees to rear queens. And they reared queen cells just fine, but from his data, those queens had a hard time. They never laid eggs. They went out for their mating flights, they came back, but they didn't lay eggs. I'll get back to that, okay? And you always want to understand that everything is spun. Every research project, every researcher wants to be able to conclude this is the most important piece of science published this year, and they usually start off with the decline of the honeybee and all that. Okay, that's spinning it. Okay, that's marketing you because you get promoted or you get a position if you get a lot of press from your findings. So understand that researchers spin it just as much as everybody else does. Okay. Oh, I showed this already. So here's the large-scale study. This is the one that just came out. This is right now. In fact, yesterday. One of the authors of this study responded to some of the criticisms, but not to this data, only to the bumblebee data. I'm waiting for the author to respond to the criticisms of this data because this essentially shows, and this chart was put, here's the problem. This was a huge monster 2-year study funded by Syngenta and Bayer. They funded it. And then these researchers published this and said, wow, we found a problem with bees. But the thing is because they funded it, they had access to all the data, not just the data that researchers chose to use. And so one of the Syngenta scientists said, well, you know what? When I analyzed the data, it looks like this. I did not see that kind of effect. And then I showed you this one where the 4 colonies in one and 5 in the other, and don't see that. So what I like to do is I like to ground truth every finding. I want to see what's happening in the real world. So I checked for ground zero, middle of the corn belt. Hey, can anybody rear queen bees with all that neonicotinoid exposure? Here's a list of all the queen producers right in the middle of the corn belt. Called some of them up. They go, yeah, our queens are fine. Okay, so when you go back to these studies that say that none of the queens lay eggs, well, these guys would not be in business raising queens if none of their queens laid eggs. It's really hard to get continuous clientele buying your queens if none of the queens lay eggs. Okay, there are some objective studies. Okay, so here's one of them. What they found is in England, even though you have all this neonic-treated canola out there, the bees don't work it that heavy. They fly to other plants around there. Now this happens there. In Canada, they work the canola really heavy, and I'll get to that in a minute. So the bees, what the effect of a neonic, it's a stimulant. So a little bit of neonicotinoid in the nectar, the bees like. If you put a little caffeine or a little nicotine in nectar, the bees prefer that to free, just like we prefer hot water with some caffeine in it to hot water without the caffeine, okay? People like a little bit of buzz, and the bees do too. They like that little bit of buzz, and they actually work harder and they rear more brood if they have a little bit of buzz. But then you increase it to a high enough level, then they start getting the shakes, and a high enough level, they shake so hard that they die, okay? So it's just a stimulant, okay? So a little bit does not seem to do much. Here's another objective study. This is very interesting. Reconciling laboratory and field assessments. So they did find a significant excess mortality on the neonic-treated bees, but the colonies appeared to be able to compensate for the excess mortality so as to preserve unaltered performance in terms of population size and honey production. And that's what we see. We're not saying that I'm not saying that the neonics are harmless to a colony, but you can feed a lot of neonic to a colony, and they compensate for it, and you don't see the effect. There was what's interesting, I was doing a search on one of the major universities' websites, and wow, here is a discussion about this paper coming into fruition between 2 major well-known researchers, and one of them had been feeding 100 parts per billion of imidacloprid to colonies over the course of the season. Now that's typically the field realistic dose is maybe like 2 to 4 parts per billion, feeding 100 parts per billion for the whole summer, and there was no effect on the colonies. So I wrote to him, I said, wow, when's that being published? And she wrote back and said, oh crap, that wasn't supposed to be public. And in a minute, it was off the web. That's never been published. That won't be published. And when I talked to researchers in Europe about what's happening, they said, oh yeah, if you want to put up proposals that I want to do some anti-neonic research, they just throw money at you. But if the results show there's no effect, you cannot get it published. You cannot get it published. And I've heard this from several researchers. Okay, so let's take a look. What's objective assessments? And the thing is, this is Paracelsus. He said, everything is poison. Water is poison. You drink enough water, you will die. I used to go into when I taught science classes, I'd come in with a 1-pound container of table salt. And I said, well, I taught small classes, typically 12, 15 kids. I said, right now I got enough poison to kill 12 kids and an adult. If we would have spread that 1 pound of table salt between us, we'd all be dead. We had a 50% chance of all of us dying. That's how toxic table salt is. Okay? Most dangerous chemical in Western society. What's the most dangerous chemical that causes the most mortality in Western society? Sucrose. Also known as sugar. More mortality due to sucrose than anything else. Okay, so what risk is toxicity times hazard. Okay, or hazard times exposure, or toxicity times exposure. If you have no exposure, you have no risk. So the question is, well, how often do you find neonicotinoids in the bee bread? Well, here's the Mullen study where they analyzed 300 or so samples, and they list every chemical down by how prevalent it was in the samples. So fluvalinate, okay, apistan, was found in 88% of the samples, coumaphos in 75%, chlorpyrifos, that's the one, the organophosphate. So the first 2 were applied by beekeepers. Amatraz was found in a large, because that was not used at that time much in the U.S. Okay? So where would the neonics be in here? What percentage of the samples would you find the neonics in? Anybody take a guess? Hey, it's killing all the bees on the planet. What percentage of the samples would there be a detection of neonics? Oh, God. It doesn't show. It's way down here at the bottom. Come on. Why won't this hide? It's 3%. 3% of the samples they found any neonics in at all. Come on. Get off. No, I want it on. I turned it off and on purpose because it's not. Okay, I guess we just cannot see that. What's that? Yeah, it's right there. It's the red right there. Anyway, 3% of the samples had any neonic residue at all. So this is the other survey. This is done every year by the USDA, and they're getting 99 samples from across the United States. Okay, and I color-coded these detects, and of course, and these also are in percent of the samples from most frequent. This is everything that was in more than 5% of the samples, 1 out of 20. The yellow ones are all beekeeper-applied miticides. The pink ones are insecticides. And clothianidin, which is the main insecticide, neonic, wasn't on the list. So I called them up and said, hey, what's the deal, guys? Why didn't you didn't test for clothianidin? She goes, oh yeah, we tested. We tested for all the neonics. We put them all together for one test. We couldn't find any neonics. And they're doing this every year. They can't find them in the samples. If there's no exposure, no matter what the hazard is, there's no risk to them. So this buffering effect just tends to take care of it. Corn, okay, maize. Exposure. Dr. Jerry Bromshank went to multiple fields. This is GLP, good laboratory practice. This is the highest quality of scientific research possibly done. What he found was 95% of the pollen samples were less than 2.8 parts per billion. 2.8 parts per billion is like drinking a cup of tea as far as an insect, okay? A little bit of stimulation, okay? Not happening there. How about comb contamination? Neonics aren't water soluble. They don't dissolve into the wax very well, so there's very little in the comb, and they don't accumulate in the combs. So the bottom line is we eat plenty of toxic chemicals, and we deal with them. So the question is, the field observations indicate that there's a problem. And some of the researchers, Norman Carrick, major European researcher, and Francis Ratnick, they've been questioning this, and we've been corresponding and challenging a lot of these papers. So Dr. Steve Shepard up in Oregon State did surveys of samples all around here, analyzed all the bee bread and wax samples, did a risk assessment. If the risk quotient is above 1, it increases, you're approaching some kind of expected toxicity. And he found essentially 99% of all the samples were below a risk quotient of 1. This has been very disappointing to a lot of the urban beekeepers who have been blaming all their problems on pesticides. When they pay for all this testing, they find out, well, no, it's not pesticides, it's that you don't control varroa. Now, there are problems. Bumblebees, bumblebee queens, when they get hit by neonics, if they're neonic plants, and a few of the species of solitary bees, not all of them. Some, like leafcutters, do fine, but osmia does not do well. So there are impacts on some of the other pollinators. That's indisputable. Benthic macroinvertebrates, a brand new study out by Sanchez Bio looking at the invertebrates that the EPA has been using to test. Didn't show much toxicity. We weren't worried about it, but these guys tested some other invertebrates, and some of them are more susceptible. So there is a question when these are used widely as seed treatments on huge acreages, and then most of the neonics bind to the soil particles, but some do get into the water. So there are questions with the food chain in rivers. So I'm hoping this evidence is used to cut back on the amount of application of the neonics. The other one is on the seed treatment, the coated seed. And the problem is with maize seed, because it's square, it grinds and some of this dust comes off, and the bees forage on the clover around where they were planting. And when I made a flow chart of the flow of pesticides to the queen, the larvae, and the pupae, I showed the dilution, how it goes from red to pink to white. It's really hard for a pesticide from outside to ever get to the queen or the larvae of the pupa. The bees do a really good job in their colony to reduce pesticide exposure because the protein, the pollen, isn't ever fed to any of the bees. The nurses do all the consumption of the pollen, and they remove the pesticide when they feed the jelly then to the bees. And you get a little tiny bee kill of the newly emerged nurses, which are pollen hogs, and some newly emerged drones, and that's it. Chemigation and irrigation and landscape uses, those can be big problems with neonics. I don't think homeowners should be allowed to use neonics at all. Okay, I was involved this one here. Bayer hired Cynthia Scott Dupre and Chris Cutler up in Canada to do the definitive study on neonics on canola. And because they wanted the results to be accepted by the beekeeping community, they invited the largest beekeeper on the planet, Brett 80, and me from the United States to go up, and beekeepers from Canada and EPA from other countries and seed treatment people. And they said, okay, before we start, we're going to lay out the whole experimental design. If you guys have any questions, tell us now so you can't criticize the results later. And Brett 80 and I said, well, yeah, there's one little thing. After the exposures, you guys are planning to put all the bees in this area, but there's a chance they might get exposed to pesticide in this area. You got to take them to somewhere where there's no other pesticide exposure. And they changed at that moment, at great expense, the whole experimental design to meet our consideration, okay? So we're really valid design. And what they found was there was no effect on the bees, on honey production, on colony strength. I showed this picture of Tim Ives in Indiana. Again, Midwest, corn and soy, no problem. And then when I'm writing these articles in the papers, I get these Canadian beekeepers writing to me, said, Randy, here's our honey production on seed-treated canola, neonic canola. He says, if this is a problem, give me more problem. This is what they do every year. These are colonies doing just fine, making a ton of hay on neonic-treated canola. Now, neonics are vastly over-applied, okay? It's not, and that constant treatment is contrary to pest-resistant management, contrary to integrated pest management. So I'm not here to sell neonics by any means. I'm an organic gardener, okay? I'm as anti-pesticide as you can get, but I'm also realistic and fair. Okay, so the future, keep this in mind. You know, we're not going to get perfection. We got 7 billion people on this planet. We got 200,000 new human beings every day. It's not going to be perfection. The best we can look for is progress on this. And what's happening is we got insects getting resistance to neonics. There's going to be new classes, okay? You can only patent for 20 years. No pesticide company plans on a pesticide lasting for more than 20 years because it goes out of patent. So they've already got the new ones coming up. There's going to be constant evolution of these pesticides. And the consumers have made it very clear to the pesticide companies, we want better, more eco-friendly pesticides. The pesticide companies have that. They know that loud and clear, okay? So work with the EPA. Support your EPAs. Donald Trump just cut our EPA's funding, okay? I am outraged. I mean, he could cut every other damn program, but keep the EPA. That's the one that protects us. This is the bottom line. This is the person right here we need to influence. The housewife who spends the dollar at the grocery store. You want to change pesticide use? She can change it in a year. If you convince the people who are buying groceries to say, I'm only going to buy groceries from farmers who do sustainable farming with minimal pesticide use, boy, in 1 year the farmers will say, you bet, you tell us what we do, you pay us to do it, and we will do whatever you want. Farmers don't like putting pesticides on. That's the last thing they want to do. As long as you pay them, they're happy to do that. So the best thing we can do is to educate the consumer to embrace sustainable agroecology. So people say, Randy, what can we do with pesticides? That's it. Educate the consumer, get them to spend their money towards sustainable agroecology. I think that's the last. Oh, there's the other one. Beekeepers. Don't piss off the farmers. You're part of agriculture, okay? The California beekeepers, there was just an anti-neonic law about to be passed. Made it all the way up to the vote in the Senate in California, and they said, well, we ought to get the California beekeepers on board as an afterthought. And the California State beekeepers said, no, we're not going to sign that one. And the environmentalists said, oh, God, why would the beekeepers stab us in the back? And the thing is, hey, we work with our farmers. We're happy being part of the agriculture community. And the last thing you want to do is get any friction between you and the agricultural community. Work with your farmers. They don't want to kill your bees. Okay. So the neonics I can't give them a clean bill of health. They're vastly overapplied, but they don't deserve, damnation either. They're just an insecticide, and if they're applied widely, they're okay. And there's a summary right there. You got any questions?
Do you have any long-term studies on neonics?
Well, how many years do you want?
How many years can you get?
Well, we've been doing a study since about 1994 in the United States of heavy-duty application, and the beekeepers, our bee numbers are going up. Canada, they've been doing the same thing in seed-treated canola. One of the major pollination contractors is Bayer Chemical, that sells canola seed, and the beekeepers happily come back to those canola plants. That's a long-term realistic. The other one, as far as like soil buildup, when I plot that out, what it does, it builds up and it reaches an asymptote and it levels out. It doesn't just keep building. You reach an equilibrium between degradation and buildup from reapplication. But when you do it mathematically, it works out with actual measured. So it's not like DDT or something like that that just keeps building up. Yeah.
Just wondering whenabouts, if this information will be published, say, on your website for public dissemination.
Oh, actually, I did have another slide. Yeah, there's a whole bunch of articles on it. Those are all on my website. If you just search for neonics, all those articles will come up. And they're not as up to date as this one. I was working on this last night and this morning, and then during break I was working on it just before I was speaking.
I'm just wondering if you've looked into fipronil, which we use for Argentinian ant bait. At a very low concentration. If it's too high a concentration, the ant will get poisoned straight away and won't take the poison back to the nest, and it's not till some time that it builds up to a high enough concentration to kill the nest. So I'm be worried about if bees are robbing a wasp nest that had just died, for example, or were able to get access to eat these baits somehow.
If
gradually
fipronil was
build up.
applied to the wasps in a sugar bait, I would be concerned, but since it's applied in a protein bait, I wouldn't expect a concern. But it's a valid concern, but it's, because it's fed in a protein bait, it's unlikely the bees would be interested.
And you don't think the other nicotinoids could build up in the woodware of the hive over time, so it could eventually get toxic, perhaps?
No, that would be very, very unlikely for that to happen, because why would they be on the woodware? And
Well,
the
if it's
bees
gradually being
don't
taken
store
out of it.
They don't store pollen or nectar on the woodware. Okay?
Fair enough. Yeah.
All right. Yeah, and the neonics are they are hydrophilic, but yeah, we haven't seen that. Like I said, we've been having this exposure since the 1990s, and we're not seeing that effect. That's reality ground truthing. All right, thank you guys. It's been a pleasure. Thank you.